Okay, activity for Chapter 3, understanding the basics of Mendelian inheritance. Hopefully most of your questions were answered during the class time, but just in case you want me to go over them, I'm also going to cover this. So the first question was looking at a pea plant with diploid number 6. And so I'm just trying to explicitly go through the process of analyzing, doing genetic analysis in these pea plants. So list all the characters and the traits. So the color of the seeds, the size of the plants, those are the two characters. So we don't specify what variant they have. This is just the general trait that we look at. And then the traits actually specify the character. So, the traits actually specify the variant of the character. So we would say a yellow seed or a green seed, a tall plant or a short plant. A cross is made between a tall yellow plant and a short green plant, and these resulted in progeny that were all tall and yellow. So assign gene names and assume that the parents came from a population that bred true. So Y and Y control color, T and T control size. So we've assigned gene names and allele names, and following the standard dominance, the capital Y indicates that it's dominant over the little y. A cross between two plants resulted in progeny that were all identical. So whenever the progeny are all identical, that's a really good hint that the parents bred true. And so for this case, we would have let me actually switch over to a text box. So we would simply have parents that were and so does this make sense with the resulting? So a tall yellow plant, a short yellow plant, and does this make sense? So the offspring of this would be genetically identical. They'd be heterozygotes, and they're tall and yellow. So basically that means that the tall is dominant and the yellow is dominant. So what are all the possible genotypes? So there's 9 total genotypes that occur, and we just listed them all out right here. You have 3 combinations of Y, 3 combinations of T, and if you multiply those together, you get 9. And so for each of these, we just use the standard dominance recessive relationships to figure out the phenotype from these. And then we also try to figure out the number of gametes. So, some of you found this relationship and posted it on Piazza. 2 to the n, so 2 to the number of heterozygote genes. And so you could use that approach, for example, to figure out the number of gametes that are produced. So this individual has 0 genes that are heterozygote, and so 2 to the 0 is 1, and so this individual produces 1. This individual has 1 gene that's heterozygote, so 2 to the 1 is 2, and we can look here. This individual has 2 genes that are heterozygote, so 2 to the 2 is 4, so this individual produces 4 total gametes, or you can simply just draw them all out in your head. To figure out that answer. So which one of these breed true? The ones that breed true are the ones that are homozygous, or the ones that only produce 1 gamete. And so there's 4 of these, and they have all 4 different combinations of the homozygotes. Monohybrid crosses. So I think a lot of you were confused at first, but I think hopefully now it all makes sense what a monohybrid cross is. So these are between individuals that are breeding true. So there's 4 individuals that breed true, and so there's 6 total combinations of ways that we can do these crosses. Actually, there's more than 6. There would be an additional crosses between the same genotypes, but they would not have any sort of genetic difference between them. So the way that the monohybrid cross works is that you would want a single trait to be different between the 2 parents. And so, if we look at this one right here and this one right here, yellow tall, yellow short, so there's only 1 trait that's different. And so we know that's an example of a monohybrid cross. If we look at this individual here, yellow tall, green tall, there's 1 trait, 1 character that's different. yellow tall, green short, there are 2 characters that are different. So this is an example of a dihybrid cross. So there's a total of 4 different ways you can do the monohybrid cross, and there's a total of 2 different ways that you can do the dihybrid cross. Sketch out all of the chromosomes at the end of prophase in mitosis of an F1 individual that results from a dihybrid cross, indicating gene and alleles on these chromosomes. So we have actually chosen to do, for this problem, we have chosen to use this particular cross that you can do, big Y, little t, cross with little y, big T, big T. And so the first thing that you need to do is think about how many chromosomes that you need to actually draw out. And so the answer to this comes from the diploid number. So you're told that the diploid number is 6. And so we know that there's 6 total chromosomes that we must have here. And so we can count 1, 2, 3, 4, 5, 6. We have 6 chromosomes to start out. So there's 2 different ways that we distinguish between them. So one is through homologous chromosomes. And we use color to distinguish between different homologous chromosomes. And then we also have to distinguish between the unique chromosomes, and that we do by length. So there's 3 unique chromosomes, so we can see that there's a large, a medium, and a small version of the chromosomes. And then since it's a diploid animal, each one of those has 2 different colors. So this would be chromosome 1, so we just follow the order is determined by the size. There's 2 chromosome 2s, and now there's 2 chromosome 3s. And each one both has a red and a blue version, one coming from the mother and one coming from the father. And so now we want to assign genes to these chromosomes. So each gene has a position, and we decided to put T on chromosome 1. And so one of the chromosome 1s has little t, and one of the chromosomes 1s has big T, big T. So some of you mixed and matched these, so you had a chromosome that had a big T and little t, and the reason that's incorrect is because you have to think about where these sister chromatids come from. So the reason that there's 2 DNA molecules in a chromosome is because DNA replication has occurred, and so whatever this allele is gets exactly copied during DNA replication. So we need to make sure that the same allele is present on each chromosome. So there's 2 DNA molecules per chromosome, so that means that there's 2 copies of each gene on a chromosome. So both of these are the same alleles, and then the opposite one has big T, big T. And then we've also chosen a position for Y, so the Y gene is on chromosome 2, and we've left chromosome 3 empty. So there's none, since we're only considering 2 genes, this chromosome doesn't have it. So it'd be an error to draw both genes on a single chromosome, and that's because of the law of independent assortment. So if these were actually on the same, if Y and T were on the same chromosome, then that would be a problem because these would no longer sort independently. So the other thing you note is that we've flipped the direction of the allele. So for the T gene, the lowercase alleles are on the red, the maternal chromosome, and then for the Y, the capital alleles are on that. And so that's actually specified by the parents. Okay, so you need to make sure that you keep this order correct. And so for this cross that we've chosen, the little t goes to this parent, which is red, and then the Y, the big Y, goes to the same parent. So we've made sure that they're both red. And then the opposite, the little y, is blue, and then the big T is blue, and that's specified by the cross. So if we did this cross instead, we would have to flip that. So red would contain both capitals and blue would contain both recessive. But because I specified the cross as this, that's basically how we drew this. So consider a pea plant now. We'll go into a test cross that resulted in 50% tall, 50% yellow, and 50% short and 50% yellow. So what is the individual genotype? And so a test cross, we always have to remember, is crossing with an individual that has recessive alleles for both. And so it allows us to determine the genotype of the parent that we're interested in. And so what we see is that in both cases, they are yellow. And so that individual must be homozygous for the yellow gene in the dominant fat way, because all of the offspring are yellow. So all of the offspring must have the heterozygote genotype. But in this case, 50% are tall and 50% are short, and so that means that the T gene must be heterozygote in the parent, because the offspring are 50% each. And so we use that information. Hopefully this is pretty easy, but we use that to figure out that the genotype is this. Now we do another test cross, 50% tall, 50% green, 50% tall, 50% yellow. And so now we notice that the color character is what's variable. All of the offspring are tall though. And so we know that means that the genotype is big T, and then for the color, it must be heterozygote because both are created. And then finally, we do a cross between these two genotypes. So if we do a Punnett square, we figure out, okay, what are the alleles that this individual can produce? So the gametes that this individual can produce, and so it can produce both a big Y, big T, or a big Y, little t. And then we draw the gametes that this individual can produce on the other side of the square, big Y, big T, little y, big T. And so we just follow this through and do the Punnett square. So there's 4 different genotypes that are produced here, and all of the offspring are yellow and tall because, if we actually figure out the phenotype of these, it's all going to be the same. All right, question number 2. We are told that we cross 2 heterozygote parents that have 3 genes that we're interested in, and you can assume that the normal, so we didn't really specify the characters or the traits, but we assume that the capitalization determines the dominance recessiveness of this. And so if we do this cross, we want to know what fraction of the progeny will share the phenotype of the first parent. And so the easiest way to do this is just to calculate the probabilities for each trait separately and then multiply them together. So you can only do this if traits are sorting independently, but we're in chapter 3, so we know that this is an assumption. So for A, what are the genotypes of A that are produced? So 1/4 will be capital A, 1/2 will be capital A, little a, and 1/4 will be little a. So this is just, following the approach of a monohybrid cross. And so there's a 3/4 chance. So these individuals will share the same phenotype as the parent one. Only this individual with a little a will not share the phenotype. So we know there's a 3/4 chance that they will share the phenotype for character 1. And you do the same thing for B, and then the same thing for C. Sorry, actually, I misspoke. So C is actually homozygous recessive, and we're asking which ones share the parent one. So C is a little bit different because in this case, 50% will be big C, little c, and 50% will be little c, and so there's only a 1/2 chance in this case. And so we simply multiply these together, 3/4, 3/4, 1/2, and that equals 9/32. You could also do this by making a giant 8x4 Punnett square. So there's 8 gametes that are produced by this individual, 4 gametes that are produced by this individual, but that obviously would be a lot more work. All right, finally, question number 3. Huntington disease is a late onset disease caused by a single dominant mutation. The following pedigree is for a family with history of Huntington's disease. And those individuals who are already suffering from the disease are shaded black. However, some additional individuals in generation 2 and 3 also have it, but because it's a late onset disease, we don't know yet whether or not they have it. So this is kind of different from the stuff, for example, we covered in the quiz, and for other traits. Most traits we assume that we can determine whether an individual has the trait very early on in life. So albinoism, for example, you can look at a child and look at the hair color, for example, to determine if they're albino. But for this disease, you don't know in late in life. So, just because an individual isn't colored now doesn't mean that they don't have it. So you're also told to assume that the individual that has the disease has this genotype. They're heterozygous, so this is a dominant mutation, so we only have to have one copy to have Huntington's, and then the wife is homozygous recessive, so the wife does not have it. So what else do we know? So the only other individual we know that's related to these individuals is this person has it, so this person must be a heterozygote, and then typically we assume that people that marry in don't have it. So this individual doesn't have it, doesn't have it, and doesn't have it. So what is the probability that individual 2C has Huntington's? So again, because this individual is marrying in, we assume that they do not have it. 2D, you have to follow the offspring and the ancestors of this individual. So this individual right here. So none of the offspring have it. So 3E through J, none of them have it yet. So we don't know for sure. So if one of these offspring had it, we would know for sure that this individual was a carrier, this individual had it, was a heterozygote. But because this is a late onset disease, the fact that they don't have it doesn't mean that this individual does not have it. And so we simply say that these progeny don't give any information about this individual's genotype, and it's completely determined by the parents. So the parents' genotype will determine the probability that this person has it, and so there's a 50% chance that this person has it because of that. What is the probability that 3K has it? So this individual, one of its parents is big H, little h. One of its parents is little h, little h. So again, it's just 50% chance if we just do the Punnett square. 3J, so 3J, what are the probabilities that 3J has it? So again, there are two possibilities for this individual 2D. So this individual is 50% chance. So when we want to look at the probability that 3J has it, basically what we're doing is a cross between, there's a 50% chance the cross is between an individual that has it with an individual that doesn't have it, and there's a 50% chance that there's a cross between an individual that doesn't have it and an individual that doesn't have it. So the probability ends up being 1/4. So there's only a 50% chance that it was a cross between an individual. And so let's kind of write it out like this. Let's see if this helps. So in this case, there's a 50% that the cross goes like this. And so if the cross is like this, there's a 50% chance that the offspring has it. And then there's a 50% chance that the cross was like this. And so in this case, there's a 0% chance that the offspring would have it. And so if we want to figure out the probability of this, we would just multiply 1/2 times 1/2 and add it to 1/2 times 0. And so the total of that equals 1/4. So the answer to that is 1/4. Similarly, 3J and 3K, if they had a child, what is the probability he or she would develop Huntington's disease? So what we want is, what we want to do here is kind of follow this same approach And so the first thing we need to do is specify the possible genotypes of the parents. So 3J and 3K. So this individual is 1/4 has it, and and 3K is 1/2 each way. So now we have to, what we want to do is actually write out all of the possible crosses. So this individual could be these two genotypes, this individual can be these two genotypes, and so that means that it could be these three different crosses. So these are the three possibilities. And so in the first case, you know, this is what happens if this individual and this individual both had it. And so that would be the first cross. And then the second one would either be this individual, the chance that this individual is this, cross with this individual that is this, or the opposite, this individual that's this is cross with this individual that's this. And then the final possibility is that both people don't have it, and so that would be this individual is this, and they cross with this individual is this. And so to figure out this, the probability of this, that would just simply be the chance that they have it. So there's a 1/4 chance that this person has it, and a 1/2 chance that this person has it. So there's a 1/8 possibility that the cross is this. In this case, there's two different ways. So there's 1/4 times 1/2 plus 1/2 times 3/4, and that's equal to 1/2. And then the final is 3/4 times 1/2 equals 3/8. So if we add these all up, there's a 1/8 possibility that crosses this, a 1/2 possibility that the cross is this, and a 3/8 possibility that the cross is this. And so now again, we just need to multiply that by the probability that that cross produces an individual with Huntington's. So in this case, it's 3/4. So there's a 1/4 chance it would be big A, and a 1/2 chance that it would be big A, little a. So there's a 3/4 possibility. And then for this one, it would be 1/2 times 1/2. So if the genotype is this, it's 1/2 times 1/2. And then the final one is 3/8 times 0. And so if we add all these together, we would get 3/32 plus 1/4, and that would equal 11/32. So the answer is 11/32. The final question, if individual 3E ends up with Huntington's, does this change the probability that 3J develops the disease? So 3E, let's look at this. So if this person has Huntington's, now all of a sudden we know that this genotype is big H, little h. And so individual D now has extra information associated with it. So this individual must have Huntington's as well, because the only way this offspring can have it is if they inherit it from one of their parents. And since we know this individual does not have it, then that means this individual now has it. And so yes, that changes the probability for 3J. And so 3J formerly had a 1/4 chance of having it. And so now that individual has a 1/2 chance of having it, because now we know that the cross is this times this, and in that case it would be a 50% chance that they pass it on. So again, this kind of follows the way pedigrees work. So you find the individuals that you know the genotype, and then you try to go up and down both both to ancestors and to offspring, and just figure out how that information propagates. So you know through inheritance rules that that also gives you information about ancestors and offspring, and that's kind of the logic behind what you follow. All right, so that is it for Chapter 3's activity.